What if we could see soft-bodied Cambrian organisms as clearly as shelled ones? In our last episode, you discovered how shells and armor evolved across dozens of lineages simultaneously. Biomineralization becoming one of evolution's most successful technologies in response to predation pressure. Trilobites built calcite exoskeletons, mollusks secreted shells, and the evolutionary arms race began. But mineralized fossils only tell part of the story. What about the soft-bodied creatures, the ones without shells or bones? This episode reveals a fossil site so exceptional it preserves what should be impossible: eyes, guts, muscles, and the strangest animals ever discovered. Fossils with eyes still visible, that shouldn't be possible. Soft tissues decompose within hours of death. Eyes, guts, muscles, gills, all the interesting parts vanish within days, leaving only bones and shells behind. That's why 99% of fossils show us armor and skeletons, but tell us almost nothing about how ancient animals actually looked or functioned. But in 1909, paleontologist Charles Walcott was hiking through the Canadian Rockies when he stumbled across something that broke every rule of fossilization: a thin layer of shale, barely noticeable among the mountain strata, containing fossils that preserved everything. Not impressions, not molds, actual soft tissues replaced by minerals so precisely that individual eye lenses were visible under magnification. Scientists initially thought it was a hoax. Soft tissue preservation this detailed simply didn't happen. The laws of chemistry said it was impossible. Yet the Burgess Shale was real, and it was about to reveal a Cambrian world far stranger than anyone imagined. But if soft tissues always decompose, how did these survive 508 million years? The answer lies not in the fossils themselves, but in how they died. The Burgess Shale wasn't a peaceful reef where animals lived and died naturally. It was a catastrophic burial ground. 508 million years ago, this site sat near the edge of an underwater cliff, a steep drop-off where shallow carbonate platforms gave way to deep ocean basins. Animals thrived on the shallow platforms above, living in what seemed like stable, productive ecosystems. Then, triggered by storms, earthquakes, or simply too much sediment accumulating at the cliff edge, the seafloor collapsed. Massive underwater mudslides swept down the slope, carrying everything in their path. Animals living peacefully moments before were torn from the seafloor, tumbled through churning sediment, and buried in deep water within minutes. This rapid burial was the first key to preservation. Entombed in fine mud, sealed from oxygen, the normal decomposition process simply couldn't start. Aerobic bacteria, the organisms that typically consume soft tissues, require oxygen. Without it, they die before they can do their work. But burial alone doesn't explain the exceptional detail. Why could scientists see individual eye lenses and gut contents? Because the sediment was extraordinarily fine, silt particles smaller than bacteria themselves. This fine mud molded around every microscopic detail before any decomposition could occur, creating perfect three-dimensional casts of tissues that would normally vanish. Over millions of years, minerals slowly replaced the organic material, preserving structures cell by cell. The result? Fossils showing not just what these animals looked like, but how they were built internally. Yet this raised an uncomfortable new question. If this kind of preservation required such specific, rare conditions, what are we missing from oceans that didn't have mudslides and fine sediment? The first creature scientists reconstructed from Burgess Shale fossils seemed like a joke. When paleontologist Harry Whittington presented his reconstruction of Opabinia at a scientific conference in 1972, the audience burst into spontaneous laughter. It was too bizarre, too alien, too utterly unlike anything alive to possibly be real. 5 eyes arranged across the top of its head. Not 2 like most animals, but 5, each on a short stalk providing nearly 360-degree vision. A long, flexible, segmented proboscis extending from the front of its head like an elephant's trunk, but lined with spines and ending in a grasping claw. A soft, segmented body with lateral lobes running down each side for swimming, a 3-pronged tail for steering. Scientists' first instinct was to assume Whittington had made a mistake, perhaps mixed up parts from different animals, or misinterpreted crushed fossils. But as more Opabinia specimens were found, all showed the same anatomy: 5 eyes, grasping proboscis, swimming lobes. Opabinia existed. It hunted successfully for millions of years in Cambrian seas. Those 5 eyes meant nothing could approach without being detected. That flexible proboscis allowed it to reach into crevices and spaces where other predators couldn't access prey. It would grab small worms or arthropods with its terminal claw, curl the proboscis backward, and deliver food to its mouth located underneath the body. But here's what made scientists uncomfortable. Opabinia worked perfectly in its time, yet left no descendants. The five-eyed body plan, so successful for so long, simply vanished. Evolution tried it, tested it, and ultimately abandoned it. Why? If a design functions well, why does evolution discard it? The answer challenges everything we assume about progress and improvement. The next Burgess creature was even more problematic. Scientists couldn't even figure out which end was the head. Hallucigenia received its name because it looked like a fever dream. For decades after its discovery, paleontologists had it reconstructed completely wrong, upside down and backward. They thought the spines were legs and the legs were tentacles on its back. They put the head where the tail should be. It wasn't stupidity. The fossils were genuinely ambiguous. Hallucigenia was so unlike anything alive that basic anatomical orientation was unclear. Only when better preserved specimens were discovered in China, showing the creature from different angles with more detail, did scientists realize their mistake. The correct anatomy: a worm-like body about 1 to 2 centimeters long, 7 pairs of clawed legs underneath for gripping surfaces, 7 pairs of long, rigid spines on top pointing upward like defensive pikes, a bulbous head at one end with a simple mouth, a tapering tail at the other end. But why would evolution create something that looks so bizarre? The answer is actually elegant: defense through confusion and deterrence. Any predator attempting to bite Hallucigenia encountered a mouthful of sharp spines. The strange appearance made it unclear which end to attack, where the vulnerable parts were, or even which way the creature was oriented. Yet like Opabinia, Hallucigenia is extinct. Its particular combination of features, effective in the Cambrian, didn't persist. The Lobopodian group it belonged to survives in a few modern velvet worms, but the specific Hallucigenia body plan vanished. This pattern was emerging. The Burgess Shale showed that most Cambrian innovations, even successful ones, failed in the long term. Success in the present guarantees nothing about the future. The most abundant Burgess Shale animal deepens this mystery: Marrella splendens, over 25,000 specimens collected from a single site. This makes it by far the most common Burgess animal, suggesting it was genuinely abundant in life, not just unusually well preserved by chance. Yet Marrella is a complete enigma. It's not a trilobite, despite superficial similarities. It's not a crustacean, though it has jointed legs and a segmented body. It's not anything that exists today. Taxonomically, Marrella is an orphan, a body plan that thrived for millions of years, then disappeared without leaving relatives. Its anatomy was distinctive: a large head shield with 2 pairs of backward sweeping spines, 1 pair short, 1 pair long and elegant. Behind the head, 24-26 body segments, each bearing a pair of jointed legs with feathery gills, large compound eyes on short stalks. No mineralized exoskeleton, just flexible organic cuticle. Marrella probably crawled along the seafloor, filter feeding on small particles or scavenging organic matter. Its abundance suggests this lifestyle was highly successful in Cambrian environments. Resources were plentiful enough to support tens of thousands of individuals in a relatively small area. Yet abundance didn't save it. When environmental conditions shifted during the Cambrian-Ordovician transition, Marrella vanished. Being numerous, well-adapted, and successful meant nothing when the rules changed. New predators appeared, new competitors evolved. Ocean chemistry altered, and Marrella, for all its success, couldn't adapt fast enough. This teaches an uncomfortable lesson. Today's winner is tomorrow's fossil. No amount of current success protects against future extinction. Some Burgess animals chose defense over everything else, and that strategy had its own problems. Wiwaxia corrugata looked like a slug that had been through an armory. Its oval body, 2 to 5 centimeters long, was covered in overlapping scales arranged in neat rows, creating flexible armor that could bend as the animal moved. Rising from this scaled surface were 2 rows of longer upright spines running lengthwise down the back, like a double mohawk. Underneath all this protection was a soft, muscular foot, just like a modern slug, allowing Wiwaxia to crawl slowly across the seafloor. Its mouth contained multiple rows of tiny teeth arranged in a radula-like structure for scraping bacteria, algae, or organic films from rocks and sediment. No eyes, no long appendages, no obvious sensory structures for detecting threats, just armor, spines, and a scraping mouth. The defensive strategy seems sound. If you're heavily armored and covered in spines, you can move slowly in the open without worrying about predators. Let them try to eat you. They'll get a mouthful of painful spines and tough scales for their trouble. But this strategy failed in the long term. Wiwaxia is extinct. Meanwhile, unarmored slugs and snails, with no spines or scales, thrive today in countless environments. Why did the armored version disappear while the naked version persisted? Because armor is expensive. Building scales and spines requires enormous energy and resources: calcium, proteins, metabolic effort. In times of plenty, this works fine. But when food becomes scarce or environmental stress increases, armored animals face a terrible choice: continue investing energy in armor and starve, or abandon armor and become vulnerable. Evolution doesn't reward perfect defense, it rewards efficiency. Sometimes being vulnerable but cheap beats being protected but expensive. Wiwaxia optimized for one strategy: armor, and that inflexibility killed it when conditions demanded different approaches. Among all these bizarre armored creatures, one was utterly unremarkable, and that insignificance is precisely why it matters. Pichia gracilans, only about 100 specimens found in the entire Burgess Shale. In a site preserving tens of thousands of Marrella, hundreds of trilobites, dozens of Opabinia, Pichia barely registers. It was small, about 4 centimeters, soft-bodied, probably filter-fed on tiny particles, and was almost certainly easy prey for any predator that bothered to hunt it. While Anomalocaris ruled as apex predator and trilobites dominated every ecological niche, from scavenger to filter feeder to predator, Pichia swam weakly through Cambrian seas, surviving in the margins, barely making an impact. Yet this unremarkable creature carried something no other Cambrian animal possessed: a notochord, a flexible rod running the length of its body providing internal support, and a dorsal nerve cord running along its back. These two features define chordates, the group that would eventually produce fish, amphibians, reptiles, birds, mammals, and humans. Every vertebrate alive today, including you, descends from something like Pichia. That humble, rare, seemingly unsuccessful creature was your ancestor. Its body plan, so unimpressive in the Cambrian, proved infinitely flexible over the next 500 million years. This reveals something profound about evolutionary success. Dominance in the present moment means nothing. Opabinia, Hallucigenia, Marrella, Wiwaxia, all successful, all abundant, or well-adapted, all extinct. Pichia, rare, weak, unremarkable, inherited the future. Success isn't about being the best right now. It's about having a body plan flexible enough to adapt to whatever comes next. The Burgess Shale's exceptional preservation reveals just how dominated by soft-bodied creatures Cambrian seas actually were. In typical fossil sites, we see mostly shells, bones, and mineralized hard parts. This creates a distorted view, making it seem like ancient oceans were dominated by armored trilobites, shelled mollusks, and calcified brachiopods. The soft-bodied majority becomes invisible, leaving no trace. But the Burgess Shale shows the truth. For every animal with a shell, there were dozens without. Soft-bodied worms, arthropods with pure organic cuticles, creatures like Opabinia and Hallucigenia with no mineralized parts at all. These dominated Cambrian ecosystems by sheer numbers and diversity. This creates an uncomfortable realization. Our understanding of ancient life is built on a tiny biased sample. We see what fossilizes easily, hard parts. Everything else vanishes unless extraordinary conditions preserve it. Even in the Burgess Shale, preservation required perfect conditions: rapid burial, fine sediment, oxygen-free environment, minimal decomposition. These conditions are incredibly rare. Most Cambrian seafloors never experience them. So what are we missing? How many soft-bodied creatures lived in environments that never fossilized? How many body plans existed that left no trace whatsoever? If the Burgess Shale required a 1 in a million preservation event, how much diversity are we blind to? The answer is deeply uncomfortable. We're reconstructing Cambrian oceans from perhaps 1% of what actually existed. The bizarre creatures we've found, the five-eyed hunters, the spiny walkers, the armored slugs, might be just the tip of an iceberg of diversity we'll never see. The Burgess Shale isn't unique, but sites like it are vanishingly rare. Only a handful of locations worldwide preserve Cambrian soft tissues with comparable detail. Chengjiang in China, slightly older than Burgess, showing even earlier Cambrian diversity. Sirius Passet in Greenland, revealing high-latitude Cambrian fauna. Burgess Shale in Australia, preserving different species in different environments. A few others scattered globally. Each site required the same perfect storm: rapid burial, fine sediment, oxygen-free conditions, and millions of years without significant geological disruption. Get any factor wrong? Too slow burial, coarse sediment, presence of oxygen, later geological folding that destroys fossils, and soft tissues vanish, leaving only shells and bones. This rarity means our Cambrian knowledge comes from a few tiny windows into specific moments in specific locations. We're seeing snapshots, not the full picture. Entire regions, entire ecosystems, entire time periods remain invisible because the right preservation conditions never occurred. And here's the truly uncomfortable part. These exceptional preservation sites reveal that normal fossil sites, showing only shells and bones, miss most of the diversity. If we applied that lesson globally, it suggests that for every known Cambrian animal, dozens more existed that we'll never discover. The Cambrian explosion was even more explosive than we can prove. The diversity we found is a minimum estimate. The true diversity was almost certainly far greater. The creatures we've discussed, Opabinia, Hallucigenia, Marrella, Wiwaxia, and dozens of others from the Burgess Shale, represent evolutionary experiments that ultimately failed. Not because they were poorly designed, not because they couldn't survive. They functioned perfectly in their time, filling niches, avoiding predators, finding food, reproducing successfully for millions of years. By any measure, they were successful organisms, yet they're extinct. No living descendants carry their body plans. The five-eyed hunters, the spiny walkers, the armored slugs, all gone. Only distant relatives in a few cases, but the specific innovations vanished. Why? Because conditions changed. The environmental parameters that made these body plans advantageous shifted. New predators appeared, new competitors evolved. Ocean chemistry altered, climate fluctuated, and these specialized, finely tuned organisms couldn't adapt fast enough. Meanwhile, a few body plans, arthropods, mollusks, chordates, proved flexible enough to persist. Not because they were superior in the Cambrian, but because they could evolve new forms when old forms became obsolete. Trilobites, while ultimately extinct, lasted 270 million years by constantly adapting. New species replacing old ones as conditions changed. Mollusks diversified into countless forms, from sessile filter feeders to active predators to land-dwelling snails. Chordates remained minor players for 100 million years before exploding into fish, then tetrapods, then everything alive on land today. Success in evolution isn't about being the best design. It's about being flexible enough to become a different design when the old one stops working. The Burgess Shale teaches a harsh truth. Perfection is temporary. Optimization for current conditions creates vulnerability to future changes. The winners are those who never fully optimize, who maintain enough flexibility to change when change becomes necessary. And that lesson applies not just to ancient creatures, but to every living thing today, including us. The Burgess Shale's strangest animals taught us that success isn't permanent, flexibility trumps optimization, and the future belongs to those who can adapt, not those who are perfect for the present. In our next episode, we'll explore those who did survive, the early chordates like Pikaia. Why did these seemingly unsuccessful organisms inherit the future, while more impressive creatures vanished? The answer reveals fundamental truths about evolutionary success that apply to every living thing, including humans. This is HistoPrimordia, where history comes alive. Thanks for watching. Subscribe now and join us for the next episode story.